can you think about a paradigm rather than you going to the lab? Can the lab come to you? And indeed, nanotechnology can make that happen. We have been working in an area called biosensors, very small miniature devices, sensors, which can sense and detect if you have particular disease and what is the severity of the disease.
So the idea is that can we use such nanostructures to detect these molecules at very sensitive concentration. If you take a typical sensor, it would measure the output from a large number of molecules. Now all those molecules are not going to have the exact same properties, and there's going to be a heterogeneity. So this is the area where we want to make devices which are able to detect just a single molecule. So a very good example would be the RNA from the virus. And we are making a particular device which is called as the nanopore sensor, which consists of a very tiny pore. That means a very small hole of the order of a few nanometers. That means the size of a single molecule. So we have this pore through which single molecules can pass through. And as they pass through the hole, we make electrical measurements and optical measurements. And through these measurements, we can actually tell which molecule is going through the pore.
Our group works on different kinds of microfluidic problems, and what we are basically looking into is to benefit from the dominance of surface tension in small scales. Again, one of the challenges in microfluidics is in droplet generation. How do you develop droplets, tiny droplets? We have developed techniques to generate droplets which are not only cost effective but very simple. Specialization of this technology is that it doesn't just print inks, it prints materials. This material jetting technology, we are trying to make it accessible to a large number of people by reducing the cost of not only establishing the setup but also to operate it. Right? And we try to address a wide variety of problems, all the way from printing of droplets to looking into separation of circulating tumor cells from patient samples. So the key technology that our group has basically contributed is to develop a mechanical way of separation where we don't use any chemical markers. And even in that mechanical way, we have tried to make the technique much more milder to the cells so that the survivability of the circulating tumor cells in the separation process improves. And the hope is that one day we will be able to, in collaboration with clinicians, provide much better treatment to cancer patients.
Another important area where nanotechnology has an impact in biology is in delivery, so what is called as drug delivery. So the idea here is that if you look at the way we treat diseases, so normally we just take some drugs and the drug molecules go inside the body and then they will do their job. An emerging theme here which is coming up is the idea of delivering drugs to the point of their need.
So what we have done in our group is to create very small vehicles of the size of about a micron or so, which can be driven with magnetic fields. These vehicles are in the form of a helical shape, similar to that of, for example, E. coli bacteria, and it moves by the action of rotating magnetic fields. So the rotating magnetic field rotates this small helix, and as a result, it can move through. We have achieved tremendously high level of control using these screws, and we are now able to move in a wide variety of different biological environments. This includes tissues containing both cancerous and normal cells. This even includes human blood, as well as inside of a living biological cell. What we have shown in this research program is that it is possible to target the cancer cells preferentially and then actually kill them at your will using external agents.
Also, we are collaborating with our friends in biology, and what we are looking into is to recreate human organs on a chip. Okay?
If you look at our program, the nanobiotechnology program at CeNSE, we see that it goes all the way from single molecule to single cell, and right now we are talking about building organs and the whole body on a chip.
Not all medicine work in a similar manner to every person. Okay? So it is very important today, and specifically in diseases where you are working with cocktail of drugs, not one drug, but multiple drugs are being given to a patient. It is very important to understand what is the response of the patient to those drugs. But what we are trying to do is to build up models of individuals on chips and look into different organs models connected together to mimic a complete body on a chip. And what it will allow us to do is that even before we give a trial drug to that person, we can test it on this platform to see its efficacy and its side effects.
So this is a product that I'm showing you, which we call Lab on Farm. About 200 kilometers from Bangalore, there is a tribal village. And that tribal village has no hospital there. They do the test instantaneously. As I said, this is a small device, that sensor that is inserted, and you get the result immediately, and the doctor prescribes the medicine.
So a characteristic feature of our Nanobio program is that we are working on problems which are likely to have a huge future impact. So we are developing the technology for the future.